record button. Let's go to the whiteboard here. All right, so I believe where we left off last time was with DNA replication. So today I would like to start with just a few little reminders of things that we have to remember about replication. First, for bacteria, it's going on almost all the time, right? So replication is practically constant, right? The cells are dividing every 20 minutes in some cases. So as soon as replication ends for one cell and the cell finishes its cell division, replication starts again in bacteria, in prokaryotes. In eukaryotes, remember, replication is only going on during the S phase of the cell cycle, which of course is, right, part of interphase, right? So we have G1, S, G2, mitosis or meiosis. So only during S phase when those chromosomes are being duplicated. In eukaryotic cells, replication takes longer, both because the machinery is slower and there's just a lot more DNA to actually replicate. Now, the next part of chapter 9, which is our focus for today, transcription. In both prokaryotes and eukaryotes, this is going on, we would say, ubiquitously or constantly, right? This is always occurring because transcription is the first step on the pathway from DNA to protein, right? So it goes DNA, RNA, messenger RNA predominantly, and that's how the cells build their proteins. So what we're going to focus on today is how we get from DNA to RNA. That's the transcription part, DNA to RNA. Now, before we talk about the steps or the process of transcription, I would like to remind you that all RNA is transcribed from the chromosome. And I know that sounds obvious at first, but I want to remind you that we're not just talking about messenger RNA, although that will be our primary focus. So we need to think about messenger RNA, transfer RNA, and ribosomal RNA, as well as what we would call microRNAs. These are predominantly regulatory RNAs and are becoming more and more a focus in microbiology because they can suppress sometimes or enhance or turn on gene expression for a particular protein. But we're going to focus on these three. Come to genetics, you will learn more about microRNAs there. Take another screen here. So types of RNA. So I would remind you first, I guess we should refresh our memories about the structure of RNA. That being that RNA has ribose as the sugar, as the monosaccharide, in the nucleic acid and the nucleotides. The nucleotides are comprised of still adenine and guanine, so still those two purines, and cytosine and no thymine, right? Instead, in RNA, no T's, right? In RNA, we have uracil instead as a replacement pyrimidine. Remember, the purines have 2 nitrogen-containing rings and the pyrimidines have 1. So there are no T's in RNA. So what happens is when the DNA has an A in it, the complement in RNA, the complementary nucleotide, is not going to be T. is going to be U for uracil. When the DNA has a G, the RNA will still, complementary will be C. When the DNA has a T on it, the RNA complementary base will still be an A. The U doesn't get replaced for both the T's and the A's, just for thymine. So I'd remind you of that. So we have a new base, uracil, and we also have a different sugar. It's not deoxyribose, it's ribose. And then, of course, there's still a phosphate group attached. Right? Now, messenger RNA, look at that one first as far as the details. Messenger RNA is pretty much single-stranded, SS for single-stranded. It is usually in the neighborhood of about, whoops. Usually in the neighborhood of about, let's say, 600 nucleotides long. Not codons, nucleotides, right? Which is going to be equivalent to 300 codons, which is long because that would be a chain of amino acids that had 300 amino acids approximately, right? So it's a big protein. Kind of give you a long list there. Single-stranded, about 600 nucleotides. It is transcribed, of course, from the DNA, and it is usually, in prokaryotes anyway, it is the sequence that will code for proteins. That part's translation. Right? The M for messenger. That was understood, but I'll put it in anyway. Messenger. Now, transfer RNA, T for transfer RNA, is a little more complicated. Transfer RNA, there are 20 different transfer RNAs, one for each amino acid. So there are 20 different transfer RNAs. Actually, there's more than that, plus. And the transfer RNA, they're not single-stranded, right? They have what we would call secondary structure. Just like with proteins, right? So I'm putting a 2 with a circle above it to indicate secondary structure. And again, you guys have seen transfer RNA before. It kind of looks like this, where it has areas where there are double-stranded. There are 3 major loops in transfer RNA. We refer to those as the loops. I would remind you that 5' to 3', it's on the 3' end where the attachment site for an amino acid is. I'll write that in yellow, amino acid attach site. Right, so this is 5' to 3' transfer RNA. It's all been folded up. And then I would also remind you that the other important part of the transfer RNA are the 3 bases down here at the bottom of the second loop or the largest loop. And we would call those bases the anticodon. And again, you already know this. This is the anticodon. That's 3 bases. They're going to be complementary to the messenger RNA codon when we talk about translation. So I don't know what that was supposed to be there, that little jiggly. Oh, 3 was the number 3. 3 bases complementary to messenger RNA. Sorry, can't get that. Can't fit it on there. I'm too close to the edge of my board. The third type of RNA ribosomal RNA often gets overlooked. This stands for little r RNA, like that, ribosomal RNA. But it's super important, okay? It's also sometimes single-stranded. Other pieces are, I should say other pieces, because it comes in 2 or 3 short little pieces. Other pieces are double-stranded. It is physically part of the ribosome, the structure of the ribosome. Physically part of the ribosome. Both the large subunit and the small subunit. And it is this ribosomal RNA was one of the first molecules that contributed to the establishment of the 3 domains for classifying living things. So bacteria, archaea, and eukarya all have distinct ribosomal RNA, and that was one of the first major molecules that helped us classify all living things into those 3 domains. Ugh. Okay, and then I'll just say briefly, like I already said, the microRNAs, they all have funny names like peewee and snurps. Those are mostly regulatory. So they deal with expressing certain proteins or interfering with certain protein expression at different times in cells. And microbiologists are super interested in them because they're hoping that you can use them to regulate or as a super specific like antibiotic to particularly inhibit a bacteria or a virus. Okay, so now we can go on and talk a little bit about the how of transcription. So transcription, that being this process of changing the language of transcribing DNA into RNA. And it is always only one of the DNA strands that is transcribed. Not both, in the same place, anyway. We call the strand that's being transcribed the template strand. So the one that's being transcribed, that equals template. And it's, for our purposes, it will be the 3' to 5' strand that's actually being transcribed, because the enzyme involved, we're not going to need all of the setup like we did for replication, because we're not copying both strands. So the main enzyme involved in transcription is a single enzyme. It's called RNA polymerase. That's not really true. There are 5 of them in E. coli and 8 of them in eukaryotes, but shh, don't tell anyone. That's, come to genetics, learn more about it. So the main enzyme is RNA polymerase, and just like DNA polymerase, it reads DNA 5' to 3', but produces new RNA, complementary and antiparallel. So it's going to produce a 5' to 3' strand, okay? Sorry, so yeah, so it reads 3 to 5. What am I doing here? It reads 3 to 5. It makes 5 to 3, sorry. 3 to 5, and it makes 5 to 3. So again, and remember the 3' to 5' strand, that's going to be what we call the template strand. Okay, how is this done? Basically 3 steps, 3-step process. We divide it into initiation, elongation, and termination. So there's a lovely picture in your textbook. It's figure 912 in that neighborhood. I'm sure it moves as well. It looks like it's made as a video as well. I'm gonna just draw, I usually don't draw transcription, but I'm just gonna make a little drawing here of transcription, and then we'll move on to translation. So the key important features to initiation involve a special sequence of DNA that we call the promoter. And one of the most famous promoters is called a Tata box, because that's its sequence, 3' to 5', right? T-A, ta-ta, T-A-T-A-A-A. This sequence is super important because it is the recognition, so it's the recognition site for RNA polymerase to know where to begin to get started. So oftentimes you'll see a picture, just like in your textbook, looks kind of like this, where we have an area of the DNA that's been opened, like this. And I did not check if I made it correctly with the correct orientation, but I'm just gonna cheat and label this the 3' or the template strand here. So here would be the promoter sequence hanging down. That's the, this is the piece we're gonna use, the template strand that we're gonna use to make our RNA. And let's make, I guess I'll make the promoter red here, right? That's gonna be the binding for our front end RNA polymerase, who is going to recognize that. So this is the RNA polymerase enzyme. All right, it's gonna bind to the promoter, and then it's going to start making complementary copy, 5' to 3', complementary, that's supposed to be a 5. So if this is an A, it's gonna put a U there. If it's a G, it's gonna put a C. And the time-lapsed picture of this, right, so the time-lapse picture of this is RNA polymerase here. And a new piece of RNA being produced. This was our original 5' piece here. Single-stranded, so those are the bases. And then you're still having complementary bases temporarily in this area. This area where the elongation is taking place, so this picture, the second picture here would already be elongation. That area has a name. We call it the transcription bubble, because that's it. The DNA is just going to be open in this spot, and we are going to transcribe from the beginning of a gene to the end of a gene. We're not copying all the DNA like we did with replication. We're not even copying all of the template strand. We're just copying, remember, if we could go back to this page, right? We're just copying about 600 nucleotides, usually less than that, okay? The termination is not super complicated. Termination is simply when RNA polymerase, and I'm going to try to draw the transcription bubble like it's moved on here. So I'm going to, like, this is the part where we started, and now the transcription bubble is further along because RNA polymerase keeps going. So here's my blue RNA polymerase again. Now my green message would be even longer. Leave it out to here. Something to do. Gives you a little mental rest. All right, so it'd still be complementary here, and this is exaggerating. In the transcription bubble, I think it's like 18 bases is it. And it is going to the end of the gene, and your book very artfully puts in a big red rectangle as the terminator, just like that. It puts like a big stop block there. So I'm just going to put terminator. And just like the promoter, it's a special sequence that inhibits RNA polymerase from continuing. You can think of it just like on a train. It's going to hit that wall up against the terminator, and when it gets there, the gene over, transcript finished. The message is going to be released, and the RNA polymerase is going to go on to do something else. So this is our mRNA here. That's supposed to be a 5, mRNA. Oh. So again, picture in your book, you can read all about it. Why do we care? Well, because we're very interested in what this green sequence is, because this green sequence is then going to be put into a ribosome so that the cell can actually produce protein. And if you remember from Bio1, proteins do everything. So the cell is constantly making protein, making all kinds of proteins, and it needs those to make enzymes, to do metabolism. That's the easiest thing to think about since we're just coming off the metabolism chapter. But anything else, right? To build new membrane, to build new peptidoglycan, whatever it is. All right, so now that brings us to translation, which is the process. I know, why do they all have to have trans in them? I don't know. It's going to get worse too because there's more trans words later. This is the process that cells use to produce protein from messenger RNA. Right. It's complicated. I like to do it in 5 steps. It requires us to know 2 things. We need to know things about the message codon, let's say, dictionary. And we also need to know things about the ribosome. Right. So the dictionary, you guys have all seen it before. And because I'm on the tablet, I can't show it to you right here, but you will find it. It's figure, I think it's figure 913 in your textbook. And I think I can just, can I just show it to you maybe? I don't know. I don't know how to make myself big. Don't know. I do know if I cut away completely to that, we'll lose all our pages. So I'm just gonna stick this up here. I don't know if you guys can actually read that or not, but looks like it's in reverse. So anyway, this is the table that you're looking for. This is the dictionary. It has all of the codons on it and then the amino acids that they code for. You will need to have a copy of that at the ready when you take this quiz, because I will ask you what amino acid is coded by ACA, and I don't expect you to have that memorized by any stretch of the imagination. I expect you to look it up in the table. If we were having it in person, I would just give you a copy of the chart, okay? Real briefly though, let's make you guys go away. I'm just gonna show you real briefly how it's laid out. You can only read messenger RNA codons from the chart. 913, okay, so yeah, figure 913. You can only read the messenger RNA codons from the chart, messenger RNA to the amino acids. And the chart is always set up the same way in every book, okay? So it goes U, C, I believe, A, G, across the top like this. But the confusing part for a lot of people is this is the second base in the codon. The first base you read from down the side, on the left-hand side. And it is also organized the same way. U, C, A, G. Look, it's making a chart. And then what never made any sense to me, even when I was a student, is they put the third base over here going down this side multiple times. U, C, A, G, and it just repeats. U, C, A, G, U, C, whoops. You get the idea. I'm not going to be able to fit all those. And how you read the chart is, for example, this first code here is going to be U, 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 because this is the first base over here on the left, the second base. This is supposed to be the third base in the codon. So this first U, 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 and U, U, C. So first base U, second base U, third base C, because you're following it over from here. Both of those code for the amino acid phenylalanine. There's going to be 4 codes in each box, 4 combinations of letters. So I'll finish this first box because that's all I can fit. So U, first base, U, second base, A, third base, that, as well as U, U, G, those both code for leucine. And the whole box is filled out. There are 64 codons. Each codon is 3 bases. Equals 1 equals a codon. And there are 3 stop codons. I'll make those red. 3 stop codons. And they mean just like it says, they mean stop. There are 2 of them in this box, UAA and UAG. And the third one is U, first base, second base, G, third base, A. Those all code stop. So I'm just going to put them in red. I'm not going to try to write stop. Yeah, so it's table 913 in your book. That's why I wrote figure 9.13. So figure 9.13. In my edition of the book, it's on page 242. I don't know if I told you guys, I did get a paper copy of the book, but it looks like it's some sort of draft copy. So I don't really know if that page number is going to be accurate. So you will need the code, this, which I refer to as the dictionary. You're going to need that to do the translating. And the translating, it might help you if you try to remember. Translating is just like translation. We're translating from one language into another. And the language, the way we're going is we're going from messenger RNA language to amino acid language, right? And the translator is going to be transfer RNA because it knows both languages. So I'm going to zip on here to a new page and we can talk about the actual translator RNA, transfer RNA portion. We already mentioned this. These 3 bases here are going to be the anticodon. And remember, up here at the top, at the 3' end, there's going to be an amino acid attached. So if in my little previous example, because I only have a few bases in my chart here, right? So if my messenger RNA, for example, which I'll make in green, if my messenger RNA along the way says U, 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 remember, we use the chart to look up the amino acids. First base U, second base U, third base U. So that one's right here. If the message says U, 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 that anticodon has to be complementary. So it's going to have a base A, because A is complementary to U, and RNA, because remember, this is transfer RNA. It's going to have A. And the amino acid attached up here, that's going to be phenylalanine, because that's what the chart tells me. come to genetics, you can learn all about the people who did the really cool experiments to figure out the chart, and how we know that this is pretty universal for pretty much all living things on the planet, which again is why we talk about evolution with a common ancestor, because all cells use this exact same mechanism in order to make their proteins. And as we keep saying, proteins do everything, and they do everything in all the cells. Okay, so here's my message. All right, so now this is a little preview, because we need to put this into context, and the context is going to be this happens in a ribosome. And it's going to be happening in 3 places at the same time. So I'm going to draw the ribosome, and I'm going to draw translation, and I believe it's written out for you. I have a handout from Bio1 that I can forward to you. Put it your put that on my to-do list. Translation handout from Bio1. Actually, it's not just translation. I have a replication translation, transcription translation handout, and it's pretty much the same as this. I add a little more detail in this class, but at least you'll have an outline of it to sort of go by when you read through. All right, as much as I like that, I am going to erase it because I'm running out of little boards. I think I'm only allowed 9, so I'm going to erase those. And I'm going to real quickly, I'm not going to write out the steps of translation. I would ask that you read those in your book and or on that outline. I can't, I'm going to have to write them out. All right, we'll just do it. Deep breath, we're almost there. All right, so let's talk about translation, the process of it. Okay. So, step 1, messenger RNA binds to the small subunit of the ribosome. First, transfer RNA with complementary anticodon to first messenger RNA codon binds to the message. Okay, let's go like this. Ribosome has 2 subunits, small subunit, large subunit. Here's my message. 5 prime, 3 prime. Messenger RNA binds the small subunit of the ribosome. First transfer RNA with complementary anticodon to the message is going to bind to that message. Let's make the transfer RNAs blue. So I'm going to put it here. Like this. There's that first transfer RNA. Here's the amino acid that's attached to it. It's already attached. That happens someplace else, right? So it has to have the correct anticodon. In most cells, this first codon on the message is always AUG. So the complementary anticodon is going to be UAC, and this first amino acid is almost always methionine, MET. All right, blah, 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 blah. Okay, step 2. So this is the drawing for step 1 and step 2. Step 2, large subunit of the ribosome binds to message. Large subunit has 3 spaces, pockets in it, and from left to right, those spaces are titled E, P, and A. So I'm going to draw this here. Here's the E site, the P site, and the A site. And in this picture, the P site is the only site that has anything in it so far. Staying here at step 2, second transfer RNA with complementary anticodon to second messenger RNA codon fills the A site. So here's my second one. It is also going to have an amino acid on it. It's going to be a different amino acid though. So I'm going to draw that one as a green circle because I can draw a square circle triangle like that. And this can be whatever it is. We could make it phenylalanine. Why don't we? Because we know that code is on the message UUU. So that would make this phenylalanine. So I'll put PAG here. All right. Next step, very important step. Peptide bond is formed between the amino acids. So this amino acid, the methionine, is going to be joined to the phenylalanine like this, that direction. I will draw it on another board here. So here's my ribosome. Here's my message bound to the small subunit of the ribosome. Here are my transfer RNAs. I know they're getting sloppy, right? Here's the large subunit. E site, P site, A site. Peptide bond, make that red because it's so important. Peptide bond. So here's my methionine from my previous picture. Oops. All right, here's the methionine. It's going to move to the top of the phenylalanine, and that's important. So here it is on top, right? There's the methionine. Here's the phenylalanine. Now, this is step 4. So this is the drawing for step 3. Step 4, ribosome moves over one codon. We call that translocating, so that empty transfer RNA number 1 is in the E site for exit. The transfer RNA, in this case, number 2 here, with the growing peptide chain, that's in the P site, and the A site, I like to say, is available, because that helps me remember that it's called the A site. Available for transfer RNA number 3. Okay, so I'll draw that real fast over here, step 4. So here's my ribosome again. Here's my messenger RNA, 5' to 3'. Now this first AUG, and I know that's UUU, and I don't know what this is, whatever. Now my AUG with my empty transfer RNA is sitting here. My transfer RNA number 2, my growing polypeptide on it, is here. And then the A site, so go back to black, EPA is open or available for transfer RNA number 3, which is going to come in here. It's going to have a different amino acid on its head, triangle amino acid. Right? It's going to have the correct complementary anticodon, and it's going to fill the A site. And step 5, go back to, I think it was step 3. Yep. Go back to step 3 and repeat. That's where the peptide bond is formed until a stop codon, and that stop codon, remember, is going to be on the message. is reached, then the peptide is cut free, an enzyme, of course, and the ribosome dissociates. I think that's associates, but I'm not sure. Yay, done. I'm gonna hit stop there. That's